Glycopeptide antibiotics potently inhibit cathepsin L in the late endosome/lysosome and block the entry of Ebola, MERS-CoV, and SARS-CoV viruses. By Nanzhou, Ting Pan, Junsong Zhang, Qianwen Li Shuizhang, Chuan Bai, Feng Huang, Tao Peng 5, Jianhua Zhang, Chao Lu, Liang Tao, and Hui Zhang. 1 Institute of Human Virology, Zhongshan School of Medicine, Sun Yat-sen University, Guangzhou, Guangdong, China. 2 Key Laboratory of Tropical Disease Control of Ministry of Education, Sun Yat-sen University, Guangzhou, Guangdong, China. 3 Guangdong Engineering Research Center for Antimicrobial Agent and Immunotechnology, Sun Yat-sen University, Guangzhou, Guangdong, China. 4 Department of Pharmacology, Zhongshan School of Medicine, Sun Yat-sen University, Guangzhou, Guangdong, China. 5 Sino-French Hoffman Institute, Guangzhou Medical University, Guangzhou, Guangdong, China. 6 Key Laboratory for Pathogenic Microbiology, CASP-MI, Institute of Microbiology, Chinese Academy of Sciences, Beijing, China. Comparative analysis further demonstrated that teicoplanin is able to block the entry of Middle East Respiratory Syndrome, MERS, virus and severe acute respiratory syndrome, SARS, virus envelope pseudotyped viruses as well. Teicoplanin derivatives such as dalbavancin, oritavancin, and telavancin can also inhibit the entry of Ebola, MERS, and SARS viruses. As the toxicity of glycopeptide antibiotics is quite low, we propose that glycopeptide antibiotics might be used in the clinic for Ebola/MERS-CoV/SARS-CoV infection, especially in the case of emergency requirements during outbreaks of these severe viral infections. Mechanistic studies showed that teicoplanin blocks Ebola virus entry by specifically inhibiting the activity of cathepsin L, opening a novel avenue for the development of additional glycopeptides as potential inhibitors of cathepsin L-dependent viruses. Notably, given that teicoplanin has routinely been used in the clinic with low toxicity, our work provides a promising prospect for the prophylaxis and treatment of Ebola, MERS, and SARS virus infection. Discussion: in our study, we performed high-throughput screening of a clinically approved drug library and identified that teicoplanin not only inhibits the entry of Ebola/HIV-1 pseudotyped viruses but also of transcription and replication-competent TRVLPS. The IC50 of teicoplanin on the entry of Ebola TRVLPS is as low as 330 nm, whereas the CC50 of teicoplanin is over 500 μm. In addition, teicoplanin inhibits the entry of Ebola viruses into different types of host cells, including primary human umbilical vein endothelial cells, A549 cells, HeLa cells, and THP1 cells. It also inhibits the entry of MERS-CoV/HIV-1 and SARS-CoV/HIV-1 pseudotyped viruses. Teicoplanin is a glycopeptide antibiotic isolated from actinoplanes Teichomyceticus. Teicoplanin contains 5 major components, 39,40, that can form complexes with the C-terminal L-lys-diala subunits of lipid 2-peptidoglycan precursors. Teicoplanin binding of lipid IIS inhibits their transglycosylation and transpeptidation, leading to disturbed cell wall synthesis in gram-positive bacteria. Recently, teicoplanin was reported to inhibit Ebola pseudovirus infection in cell culture, which is consistent with our observations. Teicoplanin was also demonstrated to have an effect on a common component used by both enveloped Ebola viruses and human respiratory syncytial viruses, but not by non-enveloped viruses. However, the mechanism by which teicoplanin inhibits the entry of Ebola remained unresolved. To elucidate the molecular mechanisms underlying this process, we compared the entry of Ebola viruses and SARS-CoVs and the effect of teicoplanin thereon. Teicoplanin represses the entry of both Ebola viruses and SARS-CoVs, which require transport to the endo/lysosomes to deliver their genomes. Since several host factors have been reported to be essential for Ebola and SARS-CoV virus infection, the identification of the common host factors that are required for the entry of both Ebola viruses and SARS-CoVs would likely be helpful to clarify the target of teicoplanin. Our data demonstrated that CTSL, VPS11, VPS18, VPS33A, VPS39, and VPS41 are all indispensable for the entry of both Ebola viruses and SARS-CoVs. The elucidation via a dextrin uptake assay that teicoplanin does not affect the functions of Hops complexes suggested that the target of teicoplanin was cathepsin L. In support of this, the results of two cathepsin L enzymatic activity assays indicated that teicoplanin indeed inhibits the activity of cathepsin L, which can explain why teicoplanin inhibited both the entry of Ebola viruses and human respiratory syncytial viruses as shown in a previous study, since cathepsin L is required for the infection mechanism of both. Considering that cathepsin L is also required for the entry of MERS-CoVs, teicoplanin thus represents a broad virus entry inhibitor. In addition, the inhibitory effects of teicoplanin and its derivatives on HIV-1, HCV, influenza viruses, flaviviruses, FIPV, and SARS-CoVs have also been reported, further supporting that the antiviral target for glycopeptide antibiotics is a common host factor. As teicoplanin can bind lipid IIS, we hypothesized that it might interact with the enzymatic domains of cathepsin L and block their functions, similar to the reported inhibitory effect of antimicrobial peptide LL37 on cathepsin L. The specific binding sites of teicoplanin on cathepsin L need to be further investigated by molecular docking, amino acid mutation, and surface plasmon resonance to confirm this hypothesis. We also examined the effects of the teicoplanin homologs dalbavancin, oritavancin, telavancin, and vancomycin on the entry of Ebola TRVLPS, MERS-CoV/HIV-1, and SARS-CoV/HIV-1 pseudotyped viruses. The data demonstrated that dalbavancin, oritavancin, and telavancin also inhibit the entry MERS-CoV/HIV-1 and SARS-CoV/HIV-1 pseudotyped viruses. However, vancomycin did not repress their infection. By comparing the structures of these compounds, we found that all the glycopeptide antibiotics that inhibit Ebola TRVLP, MERS-CoV/HIV-1, and SARS-CoV/HIV-1 pseudotyped virus entry contain hydrophobic groups at the amidogen domains of their aminosaccharides. These groups might play an important role in the interactions between the glycopeptide antibiotics and cathepsin L. In contrast, vancomycin lacks the hydrophobic groups, which might be the reason why it does not inhibit these viral infections. Fig. 9. For the treatment of gram-positive bacterial infections in the clinic, teicoplanin can be administered intravenously or intramuscularly once daily following an initial loading dose, which is convenient for outpatient therapy. Compared with vancomycin, which was the first discovered glycopeptide antibiotic, teicoplanin is better tolerated with lower nephrotoxicity. Acknowledgements. We thank Dr. Wenlin Huang, Sun Yat-sen University, for providing the pcDNA 3.1 SARS-CoV-S plasmid. We thank Dr. Lingkai Zhang, Tsinghua University, and Dr. Shivo Zhang, Fudan University, for providing the pcDNA 3.1 MERS-CoV-S plasmid. We thank Dr. Jun Li, Sun Yat-sen University, for providing the primary human umbilical vein endothelial cells, HUVECs. We thank Dr. Thomas Honen, Federal Research Institute for Animal Health, Germany, for providing several plasmids to generate TRVLPS. Conflict of interest. The authors declare that they have no conflicts of interest with the contents of this article. Author contributions. All listed authors contributed to this work and reviewed the manuscript. NZNTP, PAN, designed the experiments and performed most of these experiments. JSZ, XZ, FH, TP, Peng, TH, LT, and JHZ performed the kinds of viruses-related experiments. QWL, CB, and CL carried out the high-throughput screening of FDA-approved drug library. NZ and Hui Zhang contributed to the idea generation, experimental design, manuscript preparation, and conceived the project.